This protocol explains the fabrication of microbeads, which will be used to help control the rate of release and the amount of amyloid, which is a protein of interest. The microbeads will immobilize cells in a suitable biomaterial, and it will shelter them from their surrounding environment, as well as allowing them to exchange byproducts and nutrients with their surrounding environment. Encapsulating cells using this technique ensures tight control of microbead size as well as cell number, and we do this by adjusting various fabrication parameters.
So encapsulating the cells described in this protocol will give us more of a chronic release of amyloid to use in both in vitro and in vivo systems. And the idea would be this would give us a better understanding of the mechanisms of disease and also allow us to test new treatments.
This method can be used to formulate controlled systems and study the release of any biomolecules from any cell types, whether alone or in combination.
To begin, remove a near-confluent cell flask from the incubator. Treat the cells with 0.25% trypsin EDTA solution and incubate at 37 degrees Celsius for 5 to 10 minutes to detach the cells. After adding DMEM F12 medium, collect the cells into a 50 milliliter tube. Centrifuge the cells at 1000 RPM for 5 minutes. Remove the supernatant and resuspend the pellet in HEPI's buffered saline to double the final desired cell concentration. In a 50 milliliter centrifuge tube, mix this cell suspension in a 1:1 ratio with 4% weight and volume alginate solution to obtain a final suspension containing the desired cell concentration in a tube.